So today, after leaving kind of the cell signaling and the cell cycle, we're going to get ready to talk about uh, photosynthesis and cellular respiration, which are in kind of cell processes. Like I said, we're going to take these two kind of smaller units and put them together in what I call a cell processes unit that makes up, the cell cycle, cellular respiration, photosynthesis, and then cell signaling, those topics kind of put together. But before we get to photosynthesis and cellular respiration, we need to talk about two things. We need to talk about enzymes because enzymes are going to be super important in both of those processes, and we need to talk about metabolism or um, using chemical energy or making chemical energy in the cell to power all of the different chemical reactions that need to happen. And so today and tomorrow, we're going to look specifically at enzymes, all right? And then on Friday, you're going to look at ATP. And we're going to do that just before we start our cellular processes of cellular respiration next week. So first of all, when I say metabolism, what does that word mean? All right, a lot of us maybe are used to hearing about it in terms of somebody's metabolism is really high or really low. You know, does it seem like if they eat anything, they gain weight or they not don't? But in a scientific context, what does metabolism mean? Metabolism means it's the chemical reactions of life that keep organisms living. So that might be in forming bonds between molecules, like we talked about in dehydration and synthesis. And these are called anabolic reactions because they build larger substances out of smaller pieces. Or on the other side, there are reactions that break bonds between molecules like hydrolysis. And these are called catabolic reactions when they take larger molecules and break them into smaller ones. So I know we talked about this before in biochemistry, but just a reminder about the difference between anabolic and catabolic reactions in metabolism. So here is another example. These should be really familiar. Here is dehydration synthesis, building a larger polymer or molecule out of two smaller pieces, and water is coming out, and hydrolysis doing the opposite, breaking a larger molecule down into two smaller pieces by using water. And one thing that is interesting to look at this picture again is that we have this object called an enzyme that's necessary for this reaction to happen. And that's kind of what we're going to talk about today is enzymes' crucial role in metabolic reactions. So here's kind of the same thing, but now instead of just like simple hexagons, I replaced them with the structures of some carbohydrates. So in these images on the top, we have two monosaccharides. This happens to be glucose and fructose being put together to make a disaccharide. This is sucrose. And on the bottom, we have sucrose being going through hydrolysis to form glucose and fructose, just the opposite. So, a little bit more about chemical reactions and energy. And I'm sorry if this ends up sounding a lot like chemistry and that wasn't something that you were looking forward to, but this part will be really brief. But we're just going to turn the idea of chemical reactions into ones that release energy or require energy. So you might have heard in chemistry about classifying reactions whether they release heat, exothermic, or absorb heat, endothermic. It's going to be very similar. So chemical reactions that in total release energy are called exergonic, right? And these are the reactions in which we break more bonds than we form. These are hydrolytic, hydrolysis, catabolic reactions. So catabolic reactions are exergonic, they release energy, okay, and we're calling them exergonic. And then other chemical reactions require you to put energy into them net for them to work, and we call them endergonic. They're absorbing or requiring energy, and these are dehydration synthesis reactions. So kind of one of the foundations of all of biochemistry and studying these reactions is that in living systems, we always couple or put together exergonic and endergonic reactions. Exergonic ones need to happen first to produce the energy to allow endergonic reactions to go. So, for example, you need to break down the food that you eat and release that energy stored in those bonds to create new bonds, new chemicals that might make up the tissues of your body. So here's just a picture to represent endergonic and exergonic reactions. Um, don't start to feel too anxious about this delta G stuff that might, from chemistry, you might remember that being particularly a pain. The idea is that we want to realize that in an exergonic reaction, we end up with products that have less energy than the reactants. And in endergonic reactions, we end up with products with more energy than the reactions, a net input of energy and net output of energy. All right. Even though in both cases, kind of this hill goes up first, we are only looking at how much energy the reactants have versus how much energy the products have, as whether we're calling them endergonic or exergonic. So, kind of touching on what I said previously, where does the energy come from to allow organisms to live? And that is from, like I said, coupling together exergonic and endergonic reactions, using the energy produced by breaking bonds to power reactions that will form bonds. That's, like I said, kind of the basis of all of the biochemical reactions that happen in your body. So that comes to the question of, if these exergonic reactions go downhill, why don't they just happen all on their own? Like right now, why doesn't all of the carbohydrates in your body spontaneously, like on their own, suddenly, come apart into monosaccharides? And the main reason is because those covalent bonds between the atoms in those molecules are stable. They're in a very stable state. And so to release energy from them, we need to make them unstable. So this comes to the point of activation energy. So I told you that endergonic reactions require energy, meaning that their products have more energy than the reactants. And, exergonic reactions have products that have less energy than their reactants. Either case, to get any chemical reaction started, you need a small investment of energy. Think about it like this. If you were lighting a fire, like a campfire or something, even though you end up getting a lot of heat and energy out of the wood and once it gets going, it burns all on its own, you need to add a little bit of energy to get it started. All right? And that match that you lighted, that heat that you from the match. Or if you have, like say, like a lawnmower, right? A lot of the times to get it started, especially if it's cold, you have to prime it, right? You get some got fuel flowing through the engine, and that can kind of be pretend like that activation energy. Activation energy is what we call in chemical reactions that small bit of energy we need to kickstart a reaction to happen. All right. The idea is even when we're breaking molecules down, large biomolecules are stable. And so we have to add a little bit of energy just to get those bonds starting to break. And once they do, they'll happen all on its own. The interesting thing, though, is that because of the structure of most of the biomolecules, like carbohydrates, lipids, proteins, nucleic acids, the amount of activation energy needed to destabilize those bonds, so even in hydrolysis, it would start happening all on its own, is way too much, right? Life would never be possible, even, just to spontaneously move these reactants over the activation hill, all right? There's too much energy for life to really be possible with the activation energy that all the biomolecules need to start to break down and release the energy they have stored in their bonds. But luckily, there's a solution, and that is catalysts. So catalysts aren't just used in biological processes, they're really important in industry as well, like manufacturing. And they are substances that reduce the amount of energy that is needed to start a chemical reaction. So here's a picture of that same thing before, like right here. And if there was not a catalyst present, it would take this white upside down parabola would need to be overcome to get it to turn into products. With a catalyst, this is the activation energy that we need. So the reason that life is possible is that we have these biological catalysts in ourselves that allow the chemical reactions that need to happen to occur under less energy than they normally would. And these catalysts, when they are biological or part of living organisms, we call them enzymes. So, enzymes in our body are those things that decrease this activation energy, this energy hill that allow even exergonic reactions to happen in a timely manner. If that wasn't the case, like if you would be eating food, it would take days and days and days and days for those chemical reactions to spontaneously happen and those bonds to spontaneously break down in the food, and you'd never be able to, like, you could eat, but you'd still starve to death because it would take way too long for those bonds in your food to break down so that you could actually harness any of the energy from it. So enzymes are biological catalysts. They are all proteins and some RNA. We'll talk more about RNA later. And they facilitate chemical reactions by increasing the rate, making them happen faster by reducing the activation energy. Now, the important thing is they are not used up in the chemical reaction, so they can keep going and catalyzing reaction after reaction without being used up. And they don't change the amount of energy that we get from the chemical reaction. All right? They don't change the difference in the energy level of the reactants and the products. They just change the amount of energy we have to put in to get over that initial bump. And they are required for almost all chemical reactions in your body, which is crazy. Another thing is that they're highly specific. There are thousands and thousands of different enzymes in each and every cell controlling the reactions of life. They are very specific for what chemical reactions they can catalyze, they can work on. So each different chemical reaction will most likely need a different enzyme catalyst. All right, so when we're talking about enzymes themselves, there's three specific words that I want to talk about. One is a substrate. Now, a substrate is like the reactants, which are going to bind to the enzymes to form, this enzyme substrate complex, which is the term we use for when the reactant or the substrate is bound to the enzyme. So the substrate's the thing we're going to break apart or put together. The products are the end result of the enzyme catalyzed reaction. And then the active site is the place on the enzyme, like in this picture, these little divots, where the substrate fits in to to be worked on for the enzyme to catalyze its breaking down. So I would say to look at this picture to get a better idea about the enzyme, the substrate, the active site, and the product. Because I'm going to use these words now when we talk about enzymes the rest of today and tomorrow. So some properties of enzymes is, like I said, they're very reaction specific. So each enzyme works with a specific substrate. There's a chemical fit between the active site and the substrate, and that active site only fits certain substrates. Going back to when we talked about protein structure, this is why it's super important for proteins to be structured a certain way, because a lot of them are enzymes. And if they had a different shape, they couldn't work on the substrate that they needed to. And if they can't work on the substrate that they need to, that chemical reaction can't happen. The second important property is that they're not consumed in a reaction. A single enzyme molecule can catalyze thousands or more of reactions per second. All right? One thing I like to say is if you took your bedroom and you filled it up with hydrogen peroxide, H2O2, hydrogen peroxide will spontaneously break apart, hydrolyze into water and oxygen. But if you filled your bedroom up with hydrogen peroxide, it would take about 1,000 years for all of it to break into water and oxygen. If you put a teaspoon worth of what's called peroxidase, it catalyzes that reaction into that bedroom filled with hydrogen peroxide. All of it would be catalyzed in less than a minute. Right. Hopefully that can give you the sheer scale of how important it is that they're not consumed. And lastly, enzymes are affected by cellular conditions. So for example, anything that might denature a protein, like the temperature or the pH or the salinity, will affect the enzyme because enzymes are proteins, and if they become denatured or their shape changes, they can't work on the substrate they need to and the reaction doesn't work. Right, enzymes are kind of named for the reaction they catalyze, and you will always usually find that they end in an ASE. So if you see, most of them ends with an ACE, like sucrase and protease and lipase and polymerase, but not all of them do. Some, like pepsin, um, break down proteins and polypeptides. But most of the time, if you see it ends with an ASE, that's going to be an enzyme. All right, so let's talk about two general molecules, models of how enzymes work. And the simplest one is called the lock and key model, which means the substrate fits into a 3D activation site of an enzyme, and hydrogen bonds between the substrate and the enzyme kind of have it fit in place, kind of like a key fits in a lock. All right, so this is kind of the lock and key model. The substrate binds with, hydrogen binds with the activation site, and it fits snugly in there just like a certain key fits in a certain lock. This is the simplistic model. A little bit more accurate model is called the induced fit model. It's a little bit more accurate than that lock and key one. Just like the lock and key model, the 3D structure of the enzyme fits specifically the 3D structure of the substrate. But that hydrogen bonding between the substrate and the active site of the enzyme leads to the enzyme's shape changing slightly to fit around the substrate more tightly. This conformational change, that's what we call that shape change, brings certain chemical groups in the substrate into position so that they can be broken or that they can be catalyzed a little bit more easily. So the fitting of the substrate into the active site of the enzyme induces a shape change in the enzyme, which causes the substrate to be catalyzed more efficiently. How does this work? Well, there's a variety of different mechanisms that allow activation energy to be lowered and to speed up reactions. And one of the mechanisms is that by the enzyme binding with the substrate uh, at the active site is able to orient those substrates into a better position spatially for that reaction to occur. They bring certain substrate molecules closer together so it's, more easy for those reactions to occur, and also that the active site in binding with the substrate puts stress on the bonds in the substrate molecules that must be broken, making it easier for those molecules to separate without those bonds reforming. I don't want to get too much into this because this is like we could go really deep down the rabbit hole in this stuff, and that's very advanced biochemistry. And I'll just leave that for if you're interested or if you end up wanting to take more classes specifically on biochemistry. But anyway, that's going to be it for today. We're going to continue to talk about factors that affect enzymes and the rate of enzyme catalyzed reactions next time.